Dear students, in this unit we are going to discuss the identification of bacteria. Identification of bacteria helps us to characterize the biodiversity because the clinical samples which mostly likely contain the microorganisms, both normal flora and the pathogens. So it is important to isolate the pathogens in a pure culture using various types of selective and differential media. It is important to identify the bacteria to know how it works, in which class it belongs, its disease, and how to treat those diseases. And it is equally important to identify the bacteria obtained in a pure culture. It must be identified. A systematic stepwise procedure is adopted for identification. Following methods are based on bacterial characteristics. So first is the morphology. It depends upon number of factors like strain studied, nature of culture medium, temperature, time of incubation, age of culture, and number of subcultures it has undergone. This will be studied by microscopy after staining. In the microscopy, we can have the determination of bacterial characteristics. All the characteristics cannot be made out in a single medium and may require use of multiple media. The very first thing is shape. Now, the bacteria are having different shapes, that is oval, spherical or filamentous, rod shaped, comma shaped or spiral. Second is the size. Length and breadth may vary for each bacteria or a type of bacteria. Sides may be parallel, convex, concave or irregular. The ends may be cut straight, rounded or tapering. Considerable variation in shape and size leading to club, navicular and swollen or shadow or giant forms may be seen. Next is the motility. So bacteria may be motile or non-motile. So for identification of motility, hanging drop method is used. Next part is the arrangement of the bacteria. So they may be having different types. First involves the cocci. In cocci, there are 4 different types of arrangement. This particular arrangement is known as pair because they are in pair. Tetrads having 4 number of organisms. Then it is the chain type of structure, and this is cluster. It is having a complex structures. Next is bacilli. Bacilli will be having again different types. This is known as cocobacilli. This is fusiform bacilli, and these are miscellaneous, that is having different types of shape. And next is the spirochetes, which will be having the shape like this. Next is the spores and flagella. So flagella may be, the organism may be without flagella, that is a trichate or monotrichate, amphitrichate or peritrichate. These flagellas cannot be identified with a plain microscope. So here we have to visualize it with the help of light microscopy. This particular structure will be known as flagella. This is bacterial cell and this is flagella. So this cannot be visualized under normal microscope. So we have to go for light microscopy so that this flagella can be seen. Next part is the capsule. Capsules may be present or may not be present. It can be demonstrated by negative staining, dark ground illumination, phase contrast, or electron microscopy. The capsule can be identified. Next is spores. So spores may be present and the spores will be either oval, spherical or ellipsoidal. It may be of same width or wider than bacillary body. Spores may be equatorial, subterminal or terminal. Now these are some bacterial characteristics, and depending upon these characteristics we are going to identify the different types of bacteria. Now we will move towards simple staining. Involves the staining of bacterial cell and the cytoplasm because these both are negatively charged, and when the reagent is added onto the bacterial culture, then it gives up the hydroxide ion or accept the hydrogen ion. Due to this, the stain becomes the positively charged. After staining, bacterial cell morphology can be appreciated. For simple staining, one inoculating loop will be taken. It will be sterilized. This particular loop will be sterilized in the gas flame, and after sterilizing it, then it will be transferred to the culture which is having the bacteria, and one loopful suspension will be taken and it will be spread over the slide. Then this slide will be allowed for the air dry. Then to this fix the bacterial culture, methanol will be added so that it get fixed on the slide. Next step is addition of methylene blue. This is stain is the methylene blue, and we will allow this stain to remain for 1 minute. Then the excess of methylene blue will be rinsed through use of wash bottle, and the extra stain will be removed by rinsing the slide with a distilled water. Then particular smear will be saturated with iodine, and excess of iodine will be also gently washed, and then the slide will be placed on the microscope stage and it will be focused under 10x objective first, and then you have to select a particular area where you are supposed to focus and then use the oil immersion lens, and you are supposed to observe it under 100x power. This will be useful to differentiate the bacteria from the yeast cells. When endocervical swab culture is taken in blood agar, the staphylococcus species and yeast cell, because in the cervical area staphylococcus species and yeast both microorganisms are present. So if you have to focus which type of infection is more there, then this will be useful, and this gives, as in this particular thing, staphylococcus and yeast both are going to show the similar colonies when they are cultured in a blood agar. Simple staining is useful to isolate the bacteria from the fungi. Then it is also useful to identify the bacterial isolate. Due to their ubiquitous presence, bacillus species may present as a contaminant in the culture plate. For example, growth in the blood agar but no growth in the McConkey's agar. Identifying the shape of bacteria helps to eliminate the isolate as possible contaminants. Whatever contaminants are present over any culture of bacteria, these can be identified by having growth on blood agar or McConkey agar, so that it can identify the bacterial isolate which are as a contaminant. Whatever species are behaving as a contaminant, these can be isolated easily. Next staining is the gram staining. So we will see. This is developed by Danish physician Hans Christian Gram in 1884. It is useful for bacterial identification and taxonomic division. This procedure separates most bacteria into two groups on the basis of cell wall. Gram-positive bacteria stains purple and gram-negative bacteria stains pink. All bacteria can be detected using gram staining except bacteria which exist within host cell, for example chlamydia, bacteria lacking the cell wall, for example mycoplasma, bacteria with insufficient dimensions to be resolved by light microscopy, for example spirochetes. So these are some exceptions which cannot be detected by gram staining. Gram staining involves the following steps. First step is the fixation of clinical material by heating or using methanol. Then wash the particular material and then apply the primary stain, that is crystal violet. Then have addition of mordant or iodine solution. We have to wait for 1 minute after addition of iodine. Then decolorization is done with the help of alcohol. Wait for 10 to 15 seconds. Then use of organic solvent to remove the excess material. Then application of counter stain. Safranin will be used as a counter stain. We have to wait for 30 seconds to 1 minute and observe under microscope. Now here we can see there is a difference between cell wall of gram negative bacteria and gram positive bacteria. Gram positive bacterial cell wall contains 90% of peptidoglycan. Peptidoglycan is 90% and it is having a very thick layer. If we compare the peptidoglycan layer in gram negative bacteria, then it is quite lighter. This is very thicker one. And here uh, these are the cell wall synthesizing enzymes. This is cytoplasmic membrane. And in aqueous solution, crystal violet dissociates into Cv plus and Cl minus and it gets penetrated through this cell wall. And membrane of both gram positive and gram negative, it can be penetrated through this. But the Cv interacts with the negatively charged components of the bacterial cell and stains them purple. In both the cases it will be stained purple for gram positive as well as for gram negative. But when the iodine is added, in the next step we are supposed to add the iodine as a mordant. So this addition of iodine, that is I minus or I3 minus, is going to react with the Cv plus, that is crystal violet ions which were present here. That will be forming the crystal violet iodine complex with the cytoplasm and outer layers of the cell. Now hence it will be having its own complex. So gram negative cells, as it is having low layer of peptidoglycan, then when we are using the alcohol, then outer membrane of the cell wall from the gram negative bacteria will be lost and this peptidoglycan layer, this layer will be lost when we are adding the alcohol and hence this layer will be exposed and gram negative cells which are having thin layer, so it becomes the leaky one and the complex form, that is crystal violet and iodine complex, it will be coming out of it. It will be washed off. While that of in gram positive, this layer we can see is the very thick layer. So in this layer the CVI complex remains as it is. It will not be washed off. and in gram-positive layer, which is highly cross-linked peptidoglycan layer, it gets dehydrated. Only the water removal will take place, but the crystal violet and iodine complex will remain here only. Multi-layered culture on dehydration traps the large crystal violet iodine complexes within the cell, and hence it remains as the stained with crystal violet only. And when we are having the decolorization, then this gram positive cell remains purple, but gram negative cell, as it has been leaky, so it will be having removal of complete stain of crystal violet, and due to this again becomes non-stained. And when we are using the counter stain, then this particular will be stained again, and we will see the pink stain, and hence gram negative bacteria on staining becomes pink, while that of gram positive bacteria remains purple only. Now here we can see that what is exactly happening, that first we have taken both organisms are now similar. After, when we are taking the smear, it will be similar. Then we are supposed to flood it with the crystal violet, then use the iodine treatment, and then decolorization. Now here we can see that there is no decolorization. This is decolorized. Gram negative has been decolorized, and when we are having the flooding with safranin, so it gets stained pink, while that of it remains as that of the same. So this can be observed under the oil immersion lens using the. Now the next technique is the acid-fast staining or Zeil-Neelsen staining method. It is used to differentiate the bacteria into acid-fast staining and non-acid-fast. That is, here we can see that acid-fast staining and non-acid-fast staining bacteria. Now, if initially we are staining it with the carbol fuchsin, then both are stained pink, and afterwards we can have the separation. So this staining is useful because some bacteria like mycobacteria contains the mycolic acid in their outer membrane, which makes the cells waxy, and this waxy surface resists to the staining by having gram staining method or simple staining method. So here the carbol fuchsin, which is a primary stain, it is applied and it will be having the staining because it gets penetrated through the waxy surface of the acid fast microorganisms, and this excess of stain will be washed with the acid alcohol, and then secondary stain methylene blue will be added, so that we get the differentiation between acid fast organism and non-acid fast organisms. So procedure involves is the first step, that is it will be repetitive of the preparation of the smear, and then that smear will be placed on the slide, then fix it. Then you have to put the carbol fuchsin and keep a filter paper over it, and this will be placed on the water bath, and above that water bath you have to keep a mesh like structure, and on that mesh you will keep this slide. Then filter paper will be removed and the slide will be washed off or decolorized using the acid alcohol mixture, which is containing ethanol and hydrochloric acid. It will be used for 15 to 20 seconds. Then again rinse the slide with water, and then counter stain methylene blue is added, which will be kept for 30 seconds, and then again excess of stain will be washed out with the water, and you have to blot the slide with the bibulous paper, which is a absorbent paper. Now this particular acid fast bacteria organisms will be having bright red to intensive purple red straight or slightly curved rods. Singly or small groups may appear. For non-acids, we can have a blue color and background material should be also stained blue. So the filter paper must remain moist. There are some limitation. Here we are keeping the filter paper when we are having heating. So you have to take care that this particular filter paper should not get dried. So you have to go for addition of moisture or water so that the proper penetration of the stain occurs. Then organisms which are cultivated in the nutrient medium, so sometimes nutrient deprivation occurs, so this results in the lower lipid content and results into the poor staining. Now next step is, or next test is IMVIC test, that is indole, methyl red, Voges Prosker test and citrate test. 4 tests, that will be IMVIC test. So this is important or used for the identification or differentiation of members of the family of Enterobacteriaceae. For this particular test, the cultures have put for the growth for 24 to 48 hours at 37 degrees Celsius and tests are performed. So first test is the indole test. In this particular indole test, it is performed by using SIM medium or in the tryptophan broth or in motility urease indole medium. So SIM or MIU medium will be used. Here we can see the red ring at the top. So this indicates that a particular indole test is positive for that organism. If there is no ring, then it will be negative. Then comes is the methyl red test. Here we can see a red cherry color at the top. For negative test, there will not be any color formation. Then comes is the Voges Prosker test. There is no change of color, indicates that test is negative. If the color has been changed, then it will be positive. Then the last one is the citrate test. In this test, we can see the change of growth of the organism as well as development of the blue color, that is Prussian blue color, indicates the test is positive. This particular test is useful. So example, we can say that suppose we are performing the IMVIC test for E. coli, then it gives indole test and methyl red test positive, while that of Voges Prosker and citrate test are negative, so that we can confirm that particular organism is E. coli. Then suppose we are doing this test for Klebsiella aeruginosa, then it shows indole test and methyl red test negative, but Voges Prosker and citrate test are positive. So likewise, this test is useful for different organisms. Now we will see the next test, some biochemical tests. So first is the nitrate reduction test. This test is done by growing the bacterium for 5 days at 37 degrees Celsius in a broth containing 1% of KNO3. The test reagent consists of a mixture of equal volume of solution of sulfanilic acid and alpha-methylamine in 5 normal acetic acid mixed just before use. Then 0.1 ml of test reagent is added to the culture. A red color gets developed within few minutes, that indicates that nitrate reduction test is positive, while no color means the reaction is negative. This test is useful for the presence of enzyme which are nitrate reductase and which reduce the nitrate to nitrite. So for such organism, this test will be used. Next is the urease test. This test is done in crystalline urease method. The slope is heavily inoculated and incubated at 37 degrees Celsius. It must be examined after 4 hours and after overnight incubation. So here only the medium is important, that is Christensen's urease medium, on which it will be allowed to have the growing. Then you have to examine it after 4 hours and after overnight incubation. So the test will be negative after 4 hours of incubation and it will be positive for overnight. So urease positive culture produces a purple pink color. Urease producing bacteria are able to reduce the urea to ammonia. So because of this conversion, it will be producing the purple or pink color. Next test is the hydrogen sulfide test. So some organisms are able to decompose sulfur containing amino acids producing hydrogen sulfide among the products. When cultured in the media containing lead acetate, then they term black or brown. So instead of lead acetate, we can go for use of ferric ammonium citrate or ferrous acetate. So organisms can be grown in the culture tubes. Between the cotton plug and the tube, a filter paper strip soaked with 10% lead acetate solution is dried and it is inserted. So if the paper becomes brown, that it indicates the production of hydrogen sulfide. Next is methylene blue reduction. 1 drop of 1% aqueous methylene blue is added to the broth culture and it is incubated at 37 degrees Celsius. Complete decolorization indicates the test positive, while if the color is green, then the test is weakly negative. Next, catalase production. A loop-full hydrogen peroxide is placed on the colonies on the nutrient agar. Prompt effervescence indicates catalase production. Culture media containing the blood are unsuitable for the test as blood also contains the catalase. Next is the oxidase test or oxidase reaction. This reaction is due to cytochrome oxidase, which catalyzes the oxidation of reduced cytochrome by oxygen. A 1% to 1.5% solution of tetramethyl paraphenylenediamine hydrochloride is poured over the colonies. Oxidase positive colonies becomes maroon, purple and black in 30 seconds to 1 minute. The test can also be done by Kovacs method. That is, a strip of filter paper soaked in the oxidase reagent is placed in a petri dish and the colony to be tested is smeared on the paper in a line about 5 mm long. In reaction, the smeared area turns dark in 10 seconds. The requirement is that the solutions should be freshly prepared. Thank you.